Hello, everyone. Welcome to my video tutorial for gene and protein annotation using the biomaRt package. You can see here is the online tutorial using biomaRt to annotate gene and proteins. If you use this package in your data analysis, please acknowledge the author, share, and cite their original papers. You can read the online tutorial in details. I'm going to make a video just to cover the most important part for our data analysis. So let's go to R Studio. First, we can load the package biomaRt. Because the ensemble is the most frequently used database, we are going to focus on ensemble biomaRt database and dataset. So the first step is to identify the database you need for your analysis. We can use the function list_ensemble to display all the available ensemble biomaRt web service. So let's run the list_ensemble function. So you can see down here in the first column, we have 4 biomaRt web service database. So the genes, mouse strains, SNPs, and the regulation. So the second column is the details about the web service name and also the current version. So now you know there are 4 biomaRt web service database. So next we can use the function use_ensemble to select one of the database. For example, here we want to select the genes web service database for our analysis. We can set the argument biomaRt equals genes. So the name here should be the name in the first column, biomaRt. So let's use the gene web service for today's analysis. We can run. You can see we established a formal class marked ensemble. So let's have a look at the ensemble gene database we can run. You can see here at the moment we are using the ensemble biomaRt database. But you can see here no dataset selected. If we click the ensemble formal class mart, you can see here we have the biomaRt ensemble mart ensemble. Here is the host website. But down here you can see we don't have a selected data set. And also the filters and contributes data slot is 0. We will talk about the filters and attributes later when we use the get BM function. So in next step, we need to select a dataset for our analysis. So this is step 2. So in step 2, we can select a dataset. First, we can use the list dataset function to have a look at how many datasets are available for our analysis. Let's run list dataset function. Okay, you can see we got all the dataset. You can see here we have 214 datasets in the Gene Web Service database. We can click and have a look. You can see the first column is the dataset name. The second column is the column to show more details, the description. And the third column is the current version for the dataset. So you can see you have 214 datasets that you can use for your data analysis. But normally we use human genome. We can go further down, it is in 80. You can see here it is human gene ensemble. And the current version is the GRCH38P14. If you go down further, we can go to 107. We can find the mouse genes ensemble. You can see the current version is the CRC M39. Because we have 214 dataset here, you can also use the search datasets function to find the data set you want to use. For example, we want to use the human genes ensemble. We can search the dataset, use the pattern for human gene ensemble. Let's run. You can see down here, it shows the human gene ensemble. If you want to use the mouse gene ensemble dataset, we can search the pattern for mouse. Let's run. You can see here in the row 107, it is the mouse gene ensemble dataset. So we know the dataset name for human gene ensemble and the mouse gene ensemble. So now we can use the use dataset function to select the dataset. For example, here, if we want to use the human gene ensemble, we can set the dataset argument as the human genes ensemble, and the mat argument we set the ensemble here. Let's select the human gene ensemble. We can run. So you can see the changes here. The ensemble becomes a large mat dataset. We can click and have a look now. You can see now we have the dataset as the human gene ensembles. We also have the filters and the attributes for the human gene ensemble dataset. So if we want to use the mouse gene ensemble, we can set the dataset as the mouse gene ensemble. Let's run this code to select the mouse gene ensemble dataset. Okay, you can see the changes the ensemble dataset. It is 1.2 megabyte now. We can click and have a look. You can see now the dataset is mouse gene ensemble. And also you can see the number changes for the filters and the attributes. So you can see through two steps selection. The first step is to select the web service. You can see we select the gene web service here. And the second step is to select a dataset. So now the ensemble database is ready for us to perform our analysis. So I showed you here for two steps to select the database. This two steps procedure helps you to understand the available biomaRt web service database and also the available dataset in each web service database. Once you know the names for the database and also the dataset in each database, then you can select a biomaRt database and dataset in one step. For example, here we are going to use the use ensemble function again. We can set the biomaRt argument as genes, then dataset as human gene ensemble. Let's clear the data we established already. Then we run this one-step code. Let's run. You can see we established the ensemble database and the dataset using one-step code. We can click and have a look. You can see now the dataset is the human gene ensemble, and we have the filters and the attributes for the human gene ensemble dataset. If we want to use the mouse dataset, you can just set the dataset as the mouse gene ensemble. We can run it again. So you can see now the ensemble database changed. If we click and have a look again, you can see now the dataset is the mouse gene ensemble. And also you can see the changes for the available filters and attributes. But sometimes you may want to use a different version. Then you can select a specific version that you want to use for your data analysis. So we can use the list ensemble archives function to view available ensemble archives. Let's run. You can see down here is all the versions for the dataset. The latest version is the 110. For example, if we want to use the version 98, you can list the ensemble, use the version argument as 98. Let's run. You can see down here we have the database, genes, mouse genes, SMPs, and the regulation. Then in the second column is the version as 98. If you want to use the 98 version, then you can set the version argument as 98. For example, we want to use the version 98 for the human dataset. We can set the dataset argument as human gene ensembles, then version as 98. Let's run. So you can see now we set the ensemble database as 98 version for the human gene ensemble dataset. Okay, until this step, I hope you know how to set the ensemble database using the biomaRt package. So let's clear the database we set it here already. Then we can just set the ensemble gene database and select the dataset as the human gene ensemble. We are going to use one-step code to select the ensemble biomaRt database and the dataset. Let's run. So we will use the ensemble database and the dataset we selected here for the following analysis. So you can see the web service database is the genes, and the dataset we selected as human gene ensemble. So next we can use the getBM function to annotate the genes or proteins. So the getBM function is the primary query function in the biomaRt package. It has 4 arguments. The first one is the values. So you need to set your queries as the values argument. Then the second one is the filters argument. The filters argument are used to define the restrictions on the query. Later we will show you use different examples. And the attributes argument is the one to define the data that you are interested in retrieving. And the last argument mat, we set it as ensemble here for today's analysis. So I showed you in the ensemble database, we have the filters and the attributes data slots. So you can see here, we have a list for the human gene ensemble filters and attributes. So we can use the function list filters to look available filters in the selected dataset. Here is for the human gene ensemble dataset. Let's see the filters in this dataset using the list filters function. You can see here we have 435 filters in this dataset. We can click and have a look. You can see the first column is the name for the filters. The second column is the description for each filter. So when you perform your data analysis for human genes, you need to set the filter argument as one of the name listed here. You can also use the list attributes to look at the available attributes in the selected dataset. Let's run. You can see here in the human dataset, we have 3,169 attributes. We can click it and have a look again. You can see the first column is the name for the attributes. You can see ensemble gene ID, ensemble gene ID version. And the second column is the description. The third column is the feature page. So because we have so many filters and attributes in the dataset, you probably don't have time to look at all the names. So you can use the search filters or search attributes function to search the filters and attributes that you want to use for your data analysis. For example, we can set the pattern as ensemble ID to search the available filters in the dataset. Let's run. You can see here we found all the available names that contains ensemble and also ID. And we can use another pattern for human genome to search the attributes. Let's run. You can see we found the 3 names that you can use as attributes for your data analysis to annotate the human genome ID, human genome symbol, and the transcription names. We can use another example, set the pattern as a symbol to search the attributes. Let's run. You can see we can find the human genome symbols and also the protein names. So now, I hope you understand the 4 arguments in the getBM function. So we can use some examples to show you how to use the biomaRt package to annotate gene IDs, gene names, protein names. So the first example, I'm going to show you how to convert gene IDs to gene names. If you watched my biomaRt video tutorials, we use our biomaRt package to align the original sequencing data. But from the biomaRt package, it only gives you the gene ID. It doesn't give you the gene names. So we can annotate the gene IDs and get the gene names. So we can note the feature count from my biomaRt analysis. I saved it as R data for this demonstration. Let's double click the data and load it. So you can see we loaded the feature count data. In the data, it also has the target name. We don't need it for today's analysis. We can remove the targets. Then we can click the feature count data and have a look at the gene ID. You can see the gene ID is in the annotation data slot. Here is the gene ID. So we can get the gene ID from the feature data. Let's get it as gene IDs. You can see here we have 28,395 genes. So now we want to convert the gene IDs into gene names. So we can use the getBM function. So first we need to set the values. The values will be the gene IDs we want to convert. Then the filters argument will be the entrance gene ID because we know the biomaRt use the gene entrance ID to get the feature count data. The third argument attributes we set as gene entrance ID because we want to keep the original gene entrance ID. Then we want to get the entrance session. This is the gene name. Then the last argument is the MART. we selected the ensemble human gene dataset. So now we can use the code here to get the gene names for the gene IDs. Let's run. You can see we generated a gene names dataset. We can click and have a look. You can see the first column is the original entrance gene ID from the R subret account. Then the second column is the gene names for each gene ID. You probably notice the number difference. We have more than 28,000 gene IDs in the feature count data, but for the annotation, we only got 24,429 gene names. This is because not all the gene IDs have a gene name in our selected database. Okay, this is the first example. That's the one we frequently use to convert gene ID to gene names. Then we can run the second example. For example, if you have a free matrix array data, sometimes the array matrix data only gives you a list of free matrix identifiers. So you can use biomaRt annotation to find the gene names for the free matrix identifiers. For example, here we select 3 free matrix identifiers from the U113 plus 2 platform. So we can list 3 free matrix identifiers as the free matrix ID for the demonstration. Let's run. Now we can use the getBM function to get the gene name for the matrix IDs. So now the values will be the 3 matrix IDs. We set it here. Then the filters will be the matrix U133 plus 2 platform. Then in the attributes argument, we want to keep original ID. Then we want to get the entrance gene accession. They are the gene names. The mat argument still is the ensemble dataset we selected for the human dataset. Let's run. You can see down here we got the gene names for 3 matrix IDs. So they are GASP3, 4, and 5. As I said, we frequently use this biomaRt package to annotate gene IDs. And I already show another example here. You can use this package to get the gene names for the matrix IDs. We are going to run 3 more examples. The third example is to annotate a set of entrance gene IDs with Go annotations. For example, we want to annotate the entrance gene ID 673, 837. So we can set both IDs as entrance. Then we can use it as the values argument. For the filters, we know the entrance IDs. Then for the attributes argument, we want to keep original entrance gene ID. Then we want to get the Go IDs in the Go annotation for both genes. Let's run. You can see we generated the data named as Go IDs. We can have a look at the Go IDs. You can see here the first column is the entrance ID name. We have 2 ID 673, and also down here is the 837. So you can see all the Go IDs for both genes. Now we can run the fourth example. We are going to annotate a set of identifiers with interprotein domain identifiers. So we can select 2 mRNA sequence is the reference sequence ID. Let's set the reference sequence ID for annotation. Then the values will be the reference sequence ID. We set it here. The filters we know, they are the reference sequence mRNA. Then in the attributes argument, we want to keep the reference sequence mRNA ID. Then we want to get the interprotein ID. And also we want the third column to have a description for this protein. We can run this demonstration now. And we generated this data as the protein ID. We can click this data and have a look. You can see the first column is the reference sequence mRNA. Then the second column is the interprotein ID. The third column is the more details description for the protein. You can see the protein P53 SMAD domain. So this is the first example. Now we are going to run our last example. In this example, we want to retrieve all the entrance gene identifiers and the human Go gene symbols. For those genes have map kinase activities. So we know in Go annotation, the Go identifier for map kinase activity is Go 004707. So we can set the values as the Go identifier for the map kinase activity. Then the filters will be Go. In the attributes, we want the entrance gene ID and also the gene names in human genome. So let's run the last example to get the gene IDs and the gene names for the map kinase activities using this Go identifier. Let's run. Okay, you can see down here we have the data. The first column is the entrance gene ID, and the second column is all the map kinase name. So we finished all the examples. There are more examples on the online website. If you want to try, you can go to the last part, part 6, examples for biomaRt queries. You will find more examples. Okay, I'm going to stop from here. I hope this video tutorial could help your data analysis. Please subscribe to my channel if you haven't to do so, and share my videos with your friends. Thank you.